So we're completely switching gears. We had some presentations on clinical, and now I'm going to introduce you to manufacturing. I'm going to talk about mRNA, manufacturing, and how do we bring quality control into that. So first, a disclaimer. And the first part of my presentation will be to give you an overview on the requirements, on the analytical requirements for mRNA therapeutics. Then I will give you a short story on how Lonza and Oxford Nanopore engaged in a collaboration, and one that we believe makes a lot of sense. And finally, I will give you an overview on the manufacturing and how can these technologies simplify our workflows. So, in terms of analytical requirements, our working base came from the COVID-19 experience. So the WHO laid down some guidelines which were developed for vaccines. Okay. They told us how we should look at the template, which is a critical raw material, and it's a DNA molecule. And then they laid down the principles for the drug substance. And for that, we have to ensure things like lot-to-lot comparability. And they also told us how to do these release assays. Then finally, we have the drug product, which means encapsulated mRNA going into the vial. This has to all come together to ensure that the product is stable over time. To do that, we need to give identity, and we need to give integrity, and we need to make sure that the quality of our product is not affected through this workflow. Okay. So, with that brief introduction, how did we come together? So, it all started when, in an event here in Oxford, promoted by the Swiss Global Enterprise, where Gordon, the CEO from Oxford Nanopore, met my colleague Clemens, which is in the audience today and will be in the expert panel this afternoon. So we had a brief discussion, a couple of meetings. We laid down a few principles on how would the collaboration make sense? What would be our goals? What we would be targeting, and how could we make it different? Following that, there was a lot of background work preparation, especially between myself and Veronica, which is also here, in which we defined the scope of work, and that was then complemented by many meetings of collaboration and through collaboration, key stakeholders, and making sure that this would be a value creation for both companies. It ended up with the signature of the collaboration agreement and the public announcement. But the question here was, what are the gains for each one of us? So we had the advantage that at that time there was also the nature method that came out in 2022 as long-read sequencing being the breakthrough method. And what does this mean? For us, it means that we don't have to rely on software analysis to link the molecules. We can have the direct sequencing of DNA, but in particularly also mRNA. And this is something that is completely disruptive from everything that was being done until now, especially in a very conserved environment as it is the biopharmaceutical methods. So, and what, in my opinion, the main gains is we can have the entire molecule, but we can also look into the specific constitutive elements of mRNA. We can look into the cap, we can look into the UTRs, we can look into the coding sequence, and the poly-A. So all of these are critical quality attributes that need to be controlled, and are now being controlled by a series of methods that are orthogonal, but which require a lot of work and effort. We can also control impurities, okay, which is very important from the safety point of view. The challenge was, it's not QC ready yet. And whenever we were presenting or discussing this technology, that was the first question. But how are you going to meet these quality requirements? Okay? So the technology is already 10 years old, like Gordon mentioned, and it reached the level of maturity that, in our opinion, can now be used. Okay? There's also the preconception that, or not the preconception, but the reality that the biopharmaceutical industry, it's very conservative. It's not very prone to change. When they have something working, they don't want to fix it, okay? So if you want to bring something that it's new, even if it's better, you have some energy of activation that you need to break. What this collaboration also brought, it's the complementarity. So Oxford Nanopore has the technical know-how. We at Lonza have the know-how on how to develop a method and bring it into a submission dossier that can be then go to the authorities. So in the end, we believe that this is a win-win situation. So the objective of the collaboration is to develop a cGMP-validated method that will target multiple quality attributes. We're not only looking at the sequencing, we are also looking into the entire molecule and bringing that together. So the next few slides will be about manufacturing and how we can then bring the quality control into that manufacturing. The IVT process, it's the first step on generating mRNA. And then we go through a few steps of downstream processing to ensure that we have a clean product in the end and that we remove all the impurities. In the end, we have the drug substance, which will have to go through identity and integrity and storage. Overall, during the development, we have to ensure that integrity and identity are kept. Identity, it's in the beginning and the end. Integrity, it's throughout the entire process. So we first generate the drug substance, and then we have to encapsulate the mRNA, which is the second step. Likewise, identity, integrity need to be controlled, either throughout the process or in the beginning and in the end. So I mentioned identity, I mentioned integrity. Identity, it means that our molecule, it is what we're saying it is, in terms of mRNA and DNA, that the sequence is correct. When it comes to integrity, the picture is a little bit more complex. We have to say that there's no degradation. And just to give you an idea on how complex this task is, these are the ICH guidelines to determine what you need to achieve to make sure that your identity test is correct, and the same for integrity. Okay? In terms of a project workflow, our goal is to make sure that this technology can be used throughout the entire life cycle of the project. And that will come from R&D up to a release panel at the manufacturing. What we gain, it's a simplified method where we don't have to go through 6, 7 different technologies which have different interpretations in order to achieve the same goal. And for that, we're going to use the grid INQ as our workhorse for the entire project. So, as a summary, what we want to do is to generate a method that it's simple, that it's fast, and that gives us the quality that we need. That it's not the entire picture, because although my presentation is about mRNA, what we see is a shift into combination of therapeutical molecules. So we see mRNA, we see DNA, we see combinations. We see mRNA being used in cell and gene therapy, where any change on the sequence can lead to off-target. So having a technology that gives us the exact sequence that we're working, it's extremely important. So, and it's also important to make sure that we have the workflows simplified. And finally, I would like to say that in order to achieve what I just mentioned, we need to go into the innovation space. And the challenge is to bring that innovation space into an environment that prefers to keep innovation to when it's already very mature technology. And with that, I would like to go with the disclaimer, which I left to the end. Just to call your attention that the products are for research use only. Next year, by this time, I hope to be here, or one of my colleagues, to show you that it's no longer the case, and at least it's being used for quality control and release. Thank you so much.